Hello, thank you for joining. I'm gonna share my slides here. Let's see. Hmm, Here it goes. Thanks. So, thank you again for joining today. I'm going to talk a little bit about using Wikidata as a database for five-star linked open knowledge, specifically regarding biocreation, and more specifically, a case study of the cell type marker database, PanglaoDB. First, let's start with a bit of biocreation. So biocreation is this new term that is getting more popular within the days, and it's a techno-social field of organizing the knowledge, the information, the data that you have in the life sciences. And a big part of biocreation is to making this knowledge is structured, it's reusable, it's reused by people, and thereby treating this knowledge as data. So let's see how the knowledge of the PanglaoDB could be more used as data. So the PanglaoDB is a database of cell type markers. It's actually a database of many different things. Among of those, relations between cell types and genes that mark them. It was published in 2019. It has been widely used since then. I think it has more than 150 citations, but it doesn't mean that it can be better. I think we can take advantage of this great knowledge biocreation resources that we have and make them even better by applying semantic web skills, by applying ontologies, as you are in the Bio-Ontologies community of interest, and much more. So let's start with the five-star linked open data schema. So it was a schema introduced by Sir Tim Berners-Lee in 2010, which basically scaled linked open data from one star, which means that it's unstructured, but at least it's under an open license, to five-star. It's superstructured using the best technical standards available and fully interoperable with other content in the semantic web. We all know what one-star data looks like. It's the most common way of sharing knowledge in the sciences. It's via scientific articles. And actually not even that, because for being one-star linked open data, it needs to be open. You need to have an open license in this data set. Most databases do have open licenses, and moreover, they have this knowledge stored in a structured format. And this structured format is not only on an Excel file, but it's on TSV, on CSV, on some file format that is open and that we can widely reuse in the sciences. And that's already very good. But there are ways of technically improving this standard. One approach for five-star linked open knowledge is brought by Wikidata, which is a database from the Wikimedia Foundation that has very close links with Wikipedia and other projects like that. And technically, Wikidata is compliant with W3C standards, so it uses URLs, like URIs and URLs for concepts, which means that each concept on Wikidata has a link, a unique link that people can click and go and see what that means. It's resolvable. It can be translated to RDF, which is a standard for graph databases and can be interoperated in this sense with other resources. And it's a hub on the linked open data cloud. It's connected to many other linked open data sources, thereby making this knowledge there fully integrated with the semantic web. Besides these technical aspects of Wikidata, there are very nice social aspects of Wikidata. Anyone can edit the database and anyone can query. And that makes it very powerful and a very, let's say, interesting entry step for those that are just getting to know the semantic web world. On PanglaoDB, as I mentioned before, data is known as three-star standard. And here you see a line of the database stating that the cholinergic neurons are marked by the gene choline acetyltransferase in mus musculus and Homo sapiens, in mouse and humans. And there are a few things there that are not perfect in the sense of data quality. We have a string representing cholinergic neurons. It's not resolvable. It's ambiguous, perhaps. We have species there. They're two species in the same slot. So there are ways to improve this data in terms of data quality. And how this would look like in the five-star schema of Wikidata? Well, we would have an identifier for human cholinergic neuron, an identifier for the human choline acetyltransferase, or in this case, acetylcholinesterase gene, and an identifier for the relation between them, making a triple. A triple is this entity of a subject, a predicate, and an object that is the base of all graph management strategies. All right, so how do we do that in practice? Uh, I've set up kind of 6 steps on how to make databases integrated with Wikidata. I'm going to go in detail in each one of those, but basically it's about having the right permissions and then figuring out how this knowledge in your database fits the whole schema of knowledge on Wikidata. And then making this integration, like actually putting the data on Wikidata, and then Sparkle querying and integrating it with other resources. Well, let's go into that in detail. So first, open data. All content on Wikidata is under a CC0 license. It's public domain. It's free to reuse in any way. But then there's a limitation, right? Because the content that is ingested in Wikidata in bulk has also to be public domain. And that's a limitation because sometimes you have data as, for example, in a license that requires, it's open but requires citation. This not fits Wikidata directly, so you would need to change it to CC0. But that doesn't mean the data is not cited on Wikidata, right? Every statement on Wikidata uh, is usually followed by a reference to a regional database. It's just not, there's no legal requirement that these statement will have the reference forever. But that's just uh, details, important details, but details. In the case of PanglaoDB, there was no such license, but there was a will of the developers to make the data available. So I contacted them by email, explained what Wikidata was, what were the requirements, and I got the approval to follow up this project. And so we follow up this project. So the next step was selecting the pieces of information. In our case, was pretty clear. We wanted the cell types and the markers, and wanted to do this in a species-specific way because most of the genes are represented by species-specific identifiers. We don't actually have identifiers for multi-species genes. This is underdetermined, as far as I know. Uh, so yeah, we select these fields. And the next, we had to come up with a data schema, a data model of parsing this information into Wikidata. For that, we used relations. We used properties that were already in the database. So Wikidata has thousands of properties, which include general ontological properties, but also biomedically targeted ones. One ontological property on Wikidata is subclass of, which links a class with a higher order class. So every cell that is a human astrocyte is also an astrocyte. And so we have the subclass of relation between human astrocyte and astrocyte. And this was already possible via Wikidata. So we didn't have to create any property in that direction. But the relation between a cell type and a marker was not available there. So we proposed to the community a new property called hasMarker. And in about 3 weeks, the property was approved and we could move on with the project and the integration per se. All right. The first step in the integration per se is to match the strings on the PanglaoDB to Wikidata. For the fields that we have identifiers on PanglaoDB, this is straightforward. It's just matching the gene symbol on the database with the gene symbol on the other, and they match, and it's okay. But for the cell types, it's a bit harder. There were no species-specific cell types on PanglaoDB, nor on Wikidata. So we matched every cell type on PanglaoDB to a cell type on Wikidata manually. You search Wikidata for the concept that would match the intent of the original database. And then we created 2 subclasses of this content, of this concept, one for mice and one for humans. And these mice and human cells we connected to their markers. In total, we added about 15,000 statements to Wikidata. We used the Python package, which is the Wikidata integrator, which makes it easy to do this batch integrations with Wikidata. There was no need of a community approval. It's not a super huge change. Um, so it doesn't need any kind of, let's say, further community engagement for having it there. And that's how it looks like. So every cell type that was created in this project has now a few markers. You have a graphical user interface that you can go and see that they have markers and the genes themselves, and each statement, human coordinated neural has marker gene, is followed by a reference that says when this was retrieved, from where, and from which database. With this whole amount of information there, we can now start making very powerful queries, very powerful questions to the database. For that, we use the Sparkle Query service of Wikidata, which is available for anyone to use. If you copy this on the bottom corner of the screen, there's a URL. If you copy that, you go directly to this Sparkle query on the Wikidata query service, and you can now start playing around with that. And this is basically a Sparkle query that is asking uh, which markers of human beta cells are genetically associated to diseases, and which diseases are those. So if you run this Sparkle query, you get 46 different diseases and 26 different genes that are related to human beta cells, and you can start exploring a bit more. So this is a part of this result. So these three genes on the top, they are associated both to beta cells and obesity, thereby linking obesity and beta cells. That makes sense, right? Pancreatic beta cells, they are very involved with insulin metabolism, and insulin metabolism is known to be an important part of both obesity and type 2 diabetes. We also have a link with Parkinson's disease, and it's a not so clear how these cells are related to Parkinson's disease, but that's also a part of the magic, right? When you have these new unexpected relations, you can start building new theories about the world and asking new questions and improving the way we do science. Well, this is only linking a very tiny part of Wikidata, but with this cell type information on this bigger web of knowledge, the semantic web, we can start asking powerful questions that links proteins and uh, biological pathways and genes, and well, the sky's the limit. You can integrate any type of knowledge that is part of the sum of all human knowledge. And your database and your knowledge can also be contributed to Wikidata. So here's also an invitation for you to join this process of building Wikidata as a knowledge graph for the life sciences. And in the hopes that this also sparks a big interest for this whole field of the semantic web and ontologies, and help us building this more integrated knowledge ecosystem. With that, I finish my talk. I'd like to acknowledge all the help that this project received, both here at my University of São Paulo, at my lab, at the lab of Professor Nakaya at the University of São Paulo, where I do my PhD, and all the different collaborators on Wikidata, on this project and the others that make this tool really powerful and really used by the community. Thank you very much for your time. And if you have still some minutes, I'd be happy to answer any questions.